Hello, this is Mauricio, and this is my microbiology final exam. Um, the answer to the first question, my favorite topic was the applications of microbes and specifically food microbiology. I'm currently working towards a food science degree, and this topic applied directly to my degree. As a matter of fact, I'm currently working with a few ferments. I'm also researching into how the replacement of dairy products can affect our microbiota. And many of these concepts, or many of the concepts in this module, apply directly to my interests. And actually, they apply to some of the Ecuadorian products that I'm currently looking to work with over here in the United States. I'm trying to do certain ferments with Ecuadorian products, and so that topic applied directly to my interests. Um, Making yogurt was an activity that I enjoyed, especially when I saw the results. That was pretty cool. But I also don't want to take for granted all the valuable information that I learned in terms of aseptic procedures in the microbiology lab, washing our hands, using gloves, flaming the test tubes, paying attention to all the little details to keep our labs clean and our lab results efficient and accurate was very important too. Question number two, if I had an unknown microbe, what steps would I take to determine what type of microbe, fungi, bacteria, virus, it is, and what are the particular characteristics that I would search for? So the first step would be to take blood and stool samples in order to be able to see them under a microscope. That's the first thing to do. Then I would first try to determine whether the microbe in my samples is a prokaryote or a eukaryote. A prokaryote would not have a nucleus or any membrane-bound organelles, and it would be smaller, while eukaryotes have nucleus, have membrane-bound organelles, and are larger in size. If the microbe was a prokaryote, then I could determine that the microbe is a bacteria. If the microbe had the characteristics of a eukaryote, then I knew that it was a fungi. Or if the microbe didn't have the characteristics of a cell, then I would know that the microbe was a virus. For question number three, if I had to interact with an individual that does not think vaccines are effective, I would tell them, or I would tell that person that microbes are everywhere, and we as humans have to learn to live with them. Uh, just like we have learned about good microbes like probiotic, we have also learned about pathogenic microbes, which have affected the development of humanity. If humanity would have not learned from the existence of microbes, we would still believe in spontaneous generation, which would be absurd. Um, all this knowledge that we have, or this knowledge that we have acquired through history, needs to be applied, which is the case of vaccines. There are different kinds of vaccines. There are attenuated vaccines, which are weakened pathogens used to generate an artificial immune response um, to that pathogen with the use of memory B cells. However, if the individual is reluctant to getting an attenuated vaccine, then there are inactivated vaccines that have killed pathogens that can still have the antigens to stimulate the immune system. Um, but I would definitely mention that vaccines save millions of people from dying, and vaccines allow for millions of people to have a better quality of life than they would have if they would get infected with whatever pathogens these vaccines that we've developed protect us from. So that's the way that I would approach a person that had those thoughts. Um, for question number four, one positive relationship that humans have with microbes is the one that we have with our guts microbes. Um, since a very early age, immunity is passed on to us from our mothers through our innate immunity. However, microbes are also passed on to us. I'm not talking about pathogenic microbes. I'm talking about good microbes that don't do any harm to us. So those microbes that form part of the human's gut are the ones that help us break up most of the foods that we are not able to break up ourselves. It's obvious that advantage of this relationship are the energy that we're able to produce from those microbes breaking up the food. Um, Also having a balanced diet, because if it wasn't for those microbes, we wouldn't be able to get all the nutrition out of the fruits and vegetables, for example. However, some of the disadvantages come from not feeding our microbes a balanced diet, because they would start starving and eating our insides, um, as as scary as that sounds. Another positive relationship that humans have with microbes is that relationship that we have with Saccharomyces, Lactococcus, and Lactobacillus, um, all of which are microbes used during food processing to produce certain foods and drinks like bread, cheese, yogurt, wine, beer, and many others. Advantages of this relationship is that we have been able to maintain control, or we've been able to maintain a controlled setting for this bacteria to grow in a controllable manner in our food, and we've been able to incorporate this microbes in our products, in our food products. Disadvantage of this relationship, I would say, is not knowing about enough microbes yet. So uh, we know about probiotics and this and that, and the yeast in beer, and and but there are millions of microbes that we don't know yet about. So that's a disadvantage. I think that the same relationship that humans have with microbes used to produce foods and drinks can become a negative relationship when pathogenic microbes invade what we consume, and then they spread, getting us sick, and in some cases even dying. The advantage from this relationship is that we can always study these pathogens or these pathogenic microbes, and our medicine advances fast as well and is able to study cures against those pathogens and combat them through antibiotics and vaccines. Disadvantage of this relationship are unknown microbes for which we haven't developed cures yet and can still affect the world as a whole, as what happened with COVID-19, for example. And one last negative relationship that humans have with microbes that I will mention is when microbes infect the soil or interact with the environment and get passed on to our crops. These microbes can kill many of our crops. They can generate huge losses and food shortages. And again, an advantage of this relationship is that we quickly develop pesticides and ways to control these microbes. But other problems surge from this, like toxicity in the products that we use, and all kinds of new problems can arise as well. Disadvantages are the contamination of the planet and also the ability of microbes to mutate and keep attacking our crops and keep finding ways to attack. And for question number five, I'm gonna talk about five uses of microbes. Um, so first, microbes are present in low nutrient environments, specifically in aqueous environments like lakes or rivers. And the growth of microbes in these environments is typically through biofilms, and these microbes are usually very efficient in the way they absorb nutrients, and they help us study nutrient absorption. A second use of microbes is that microbes present in terrestrial habitats help with the creation of antibiotics. A third use is that microbes that are decomposers are part of the carbon cycle, and there are also microbes that are part of the nitrogen cycle and the sulfur cycle. Fourth use for microbes is that microbes are used in wastewater treatment. And finally, microbes are used in the food industry, for example, in fermentation processes, which I talked about in this final exam. So that is it. Thank you very much. I enjoyed this class very much. Thank you for everything.